Hey guys, this is my exam 2 review for microbiology. So I realized that gram-positives and gram-negatives are more on this test than they were on last test, so I'm going to briefly go over that. But I am going to refer you back to my prior video so that you can learn a little bit more about gram-positives and negatives and how the gram staining procedure works. However, I've got a couple notes here. You guys probably can't see them. Don't worry, they're for me. We're going to go over enzymes, prokaryotic versus eukaryotic cells, some of the cell parts, active versus passive transport, and tonicity. And then we're going to do a brief overview of respiration. I'm not going to go over it entirely just because I have a really good video that I would prefer you guys watch, because I know that if I were to be the one teaching it, I would probably butcher it. So that link should be below. I will be putting that in as soon as I post this video. So, with enzymes, I've got a kind of crappy one drawn here, but enzymes are proteins, and that's a really big thing to understand, is just that they're proteins. So after a whole series of things of protein like synthesis and folding and whatever, you're going to end up getting your enzyme. And what they do is they speed up reactions. So the best way that she's ever described it, Dr. Bashi, is that they almost have like little arms that'll protrude to go grab like the substrate so that reactions can occur faster. And they kind of just act as a catalyst. So if you've taken chemistry, that term shouldn't be unfamiliar to you. To denature a protein, either high temperature, extreme changes in pH, or salt concentration are going to be what denatures the protein. And just makes it so it can't work, either by changing shape or destroying it. So on your enzyme, you're going to have this protein portion, which is going to be half of your enzyme. Then you're going to have your other part of your enzyme, which is going to be something probably like a K+ ion. It just kind of activates it, and that'll bind to some part of it. And then you're going to have this active site here. So what happens is you're going to have a substrate that's going to be floating along, and I'm going to butcher this shape, but basically you're going to have, and you can't see that one. So once you get your your protein portion and your ion portion that activates your protein, you have what we call a hollow enzyme. And then from there, you'll have a, this one should be, yeah, okay. So then you're going to have a substrate, and that substrate is going to come into this active site here so that it can complete the reaction. Some enzymes work by just grabbing all of the substrates so that they're more easily accessible. Some actually perform the reaction, but in the grand scheme of things, they speed up reactions and make them occur faster. So with that being said, we've got competitive versus non-competitive inhibition. So with competitive inhibition, you're going to have another substrate that's going to bind at the same active site. They're going to be competing for that same active site. So if the substrate, so like this is your substrate A, which is what's supposed to go in there, with your substrate B, which is what's going to block the enzyme from doing its job. If that binds first, then until that's released, your substrate A can't bind, meaning that the reaction won't ever occur. However, we also have non-competitive inhibition, which is where you have there's like another spot over here, and you've got a substrate that will come in and bind to this site, which is called the allosteric site, and they'll bind right there, and what will happen is it'll end up changing the shape of the active site so that the substrate can no longer bind to it. And so that one's non-competitive because it's not competing for the same active site, whereas the other one that I mentioned first, where they're both the substrate and whatever the blocker is are coming into the same site, that would be your competitive inhibition. that's honestly all I feel. Um, that's the general gist of enzymes that she talked about. So now I want to go a bit more into cell parts. She handed out, she had you guys fill out this paper. Mine might have a couple more on it than your guys' do, just because it's 2 years old. But I would suggest making flashcards. For this test, if you want to make flashcards, I would suggest making it on your cell parts and on some of the respiration processes, just understanding the difference in between each step. However, I do want to go over a couple of these cell parts, but I want to get into prokaryotic versus eukaryotic first. So your prokaryotic cells are kind of boring. We do a lot of research on them because they're easily manipulated, but for the most part, they're kind of boring in comparison to eukaryotic cells. So we'll have, let's draw a bacillus, and then we'll draw like a eukaryotic cell over here. Okay, so we'll have our prokaryotic here. And we'll have our eukaryotic right here. Okay, so a big difference in these is where the DNA is located. Within your prokaryotic cell, you're going to have what we call a nucleoid region, which means all of the DNA is going to end up in one area of the cell, whereas in a eukaryotic cell, you're going to have what we call a nucleus. Pretty simple, right? All of the DNA is going to be tightly wound and compacted in here, and then in your prokaryotic, you're going to have your DNA just shoved into like a little compartment, like a little area of that cell. Prokaryotic and eukaryotic cells are both going to have ribosomes as well. So I'll draw those in red because that should show up. Okay, so there we've got our ribosomes. Ribosomes are important in protein synthesis, which is why they're needed in both prokaryotic and eukaryotic cells, because it's important in DNA replication or DNA transcription and making proteins. It's important in all the cell processes that the cell needs. So you're going to have it in both because both have to be able to produce proteins. From there, this is when it starts getting a little different. They both have different means of movement. You're going to have flagella in both eukaryotic and prokaryotic cells. The biggest, easiest way for me to remember this, because at first I thought it was only in prokaryotic, is in sperm. They do have a flagella on them, and sperm is in eukaryotic cells and eukaryotic organisms. So understanding both of them will have flagella is super important. Another one, I guess from here we just kind of look at the differences in them. So you're going to have cilia in eukaryotic cells, and I'll draw them on our eukaryotic cell here. Let me do it in purple. So they're going to be these little, like, tiny hairs surrounding the cell. And what happens is they kind of just beat together, like, in order for it to move, they kind of have to like move, like, they have to beat together. That's the best way to describe it. They're gonna like have the same movement to help push it throughout the whatever fluid they're in or wherever they're living to help kind of spread it around and help it move. In prokaryotic cells, there's a couple differences, at least with locomotion. The only one being we have our spirochetes, and I talked about this a lot more in the last review, where we've got our coccus shape, which is going to be circular, and then we'll have our bacillus shape, which is going to be rod-shaped, and then we'll have our spirochetes or spirillum, which basically kind of just look like a curly fry, like that. So your axial filaments are mostly going to be found in your spirochetes or spirillum or whatever these circular ones are. and not circular ones, the spiral ones are. And what they do is they kind of like contract and relax like a muscle would to help it squiggle around. So if you were to look at a spirulina moving, it's really cool. I suggest you look it up. It like wiggles throughout the, like whatever fluid it's in or wherever it's at to get to from point A to point B. And it's just from those axial filaments contracting and relaxing just like a muscle would. And then another distinction with like filaments or like attachment things. With your prokaryotic cells, you're going to have what we call fimbriae and you're going to have a pilus. Sometimes. It depends on the particular cell, but these are specifically prokaryotic traits. So fimbriae is going to help with attachment and adherence. The best way I can describe this one is if you've ever gone hiking or horseback riding and you go to fields with a whole bunch of burrs, you get them stuck to you everywhere. And they're a pain in the neck and you have to pull them off. But fimbriae work the same way. So like the little hooks that they have on the burrs, that's kind of a lot like what fimbriae are, is they're little filaments that help to attach to things, just like the hooks on a burr. And then with your pilus, that's going to help with spreading information. So we talked a little bit about like conjugation and what a sex pilus is. That's basically what it is you have some filament that goes from one bacteria to the next to help spread DNA or information from cell A to cell B. And that's about it on that one. So I want to go a little bit more over cell parts. Like I said, I'm not going to go super like in depth into it, but I would suggest making flashcards for it. I feel like that would really help. If you're not someone that learns through like memorization super well, I'm going to try to make this review perfect and make it awesome for you so you understand a little bit better. But the key ones that she talked about are your smooth endoplasmic reticulum, your rough endoplasmic reticulum, Golgi apparatus, and your lysosomes, and I think there was one more. Mitochondria, I feel like we understand, but I'll go a little bit into that one. Yeah, okay. So, I like to kind of relate them to something that I understand super well. So I try to give all of them jobs. Which is weird, but bear with me. So with your endoplasmic reticulum, whether it's smooth or rough, both of them are going to synthesize or make phospholipids. We learned a little bit more about lipids, carbohydrates, all your macromolecules last test, and that's where this comes in. So your rough endoplasmic reticulum and your smooth endoplasmic reticulum are in charge of synthesizing phospholipids, which are used in your plasma membranes, so like your nuclear membrane or your like entire cell membrane, to help with making it so things don't spread around super easy and making it so that the cell can actually survive. The difference between your rough ER and your smooth ER, it's kind of in the name, is one is rough and one is smooth. The difference is that you have ribosomes on your rough ER and you don't have any on your smooth ER. Now, ribosomes, like I mentioned before, are used for protein synthesis. So it's kind of implied and understood that in your rough ER, you're going to be synthesizing proteins. In your smooth ER, however, you end up synthesizing steroids, which is another lipid we talked about. So the way I remember this is smooth and steroid both start with S. It's kind of a silly way to go about it, but if you can understand the process of like understanding that ribosomes do help make proteins, then that kind of takes care of the rough ER for you. And then smooth ER, just understanding steroids and smooth both start with S, so that's my way of remembering it. Then we've got our lysosomes. So lysosomes basically will just end up ingesting whatever like cell debris is in the cell, parts of dead bacteria, things that aren't necessarily needed in the cell anymore that are able to be broken down, and they dissolve them with all the enzymes inside of them. The way I like to think about this one is that it's the garbage can. So like you'll throw all your stuff in the garbage can. His job is to come pick it up and deliver it to the landfill, which can then be dissolved and broken down. So there's that one. So we covered our endoplasmic reticulums, both smooth and rough, and we've talked about our lysosomes. Next, let's talk, and we talked about ribosomes as well. So next, let's talk about our Golgi apparatus. So our Golgi apparatus is a series of membranes within the cell, and it looks really cool. If you've ever seen it, it kind of just like is this little squiggly thing throughout it. And both your endoplasmic reticulum and your Golgi apparatus are this giant series of membranes within your cell. And the way that I've always remembered the Golgi apparatus is its Amazon, basically. Is it figures out what something in the cell wants or something outside of the cell wants, and it packages it and sends it to where it needs to go. So UPS, Amazon, you get the point. It just kind of takes what needs to be used or whatever's been synthesized and packages it and sends it to different parts of the cell or outside of the cell. With that being said, when I say package, it puts it in its own little plasma membrane, using the phospholipids made in both the smooth and the rough endoplasmic reticulum, which is why I said this series of membranes throughout the cell is super important. Okay. That was a brief overview of cell parts. I mean, on your paper, it goes over stuff like chromosomes, nucleus. I feel like nucleus is super self-explanatory. It's just this plasma membrane surrounding all your DNA within your eukaryotic cells. Mitochondria, we've learned this since 7th grade. It's the powerhouse of the cell. It's what helps with making energy, and we learn a lot more about that in the respiration chapter, which is why I haven't really talked about it, because I'm going to get to that one, I promise. And then, yeah, that's about it. This part does talk, this sheet does talk a little bit about your cell processes. So I'm going to talk a little bit about that, like your endo, your endo and exocytosis, and your phago, and receptor-mediated endocytosis. But I wanted to get that out of there. So, okay. So, with that being said, let's talk a little bit about endo and exocytosis. So, endo and exocytosis both start with E, so assume it's only eukaryotic. In this class, it's only eukaryotic. You're going to see it in like your white blood cells and other immune cells throughout your body. And so what endocytosis is, it's just bringing things into the cell, and exocytosis is pushing things out of the cell. And this process is really, really cool, so I kind of wanted to go over it just a little bit. You're going to have at least with exocytosis, at top level, okay. So you've got your exocytosis, and then we're going to have, I'll do it in a second. You'll have your endocytosis. So, here's what's going to happen with exocytosis. You're pushing something out of the cell because you don't need it anymore, or it needs to go somewhere else, whatever. So you're going to have whatever is needed within this little vesicle that's being formed. Okay. And a vesicle is just basically a little plasma membrane surrounding stuff. So what's going to happen is whatever this is, like let's say it's proteins and your cell doesn't need it anymore. They're going to go into, they're going to come up to the surface of the cell membrane and they're going to start pushing it out, which is why I formed this little bubble here. It pushes against the cell membrane and the cell membrane will come together and it'll push it off and release it. So your plasma membrane is pretty movable in the sense that it can push vesicles out and take vesicles in as it wants, and then as soon as that vesicle leaves, the plasma membrane will push itself back together, or as soon as a vesicle comes in, this plasma membrane will attach itself to the it'll attach itself to the original plasma membrane, so you have more phospholipids within your plasma membrane. So exocytosis, pushing something out. With endocytosis, what happens is you're going to have these molecules out here, and they need to go into the cell. They're going to they're going to push against this plasma membrane to form a vesicle, which will then at some point pinch off here and come in. And this plasma membrane will at some point be added back into it. And that's just to bring things in. But with endocytosis, we do have 3 different types, which is the next part. So, with endocytosis, we can have receptor-mediated endocytosis, we can have phagocytosis, or we can have penocytosis. So, penocytosis, last review I had, one of the students mentioned that, because I was like, I don't have a way to remember this. She was like, it's like drinking a pint. So, penocytosis is drinking. You're drinking extracellular fluid to get whatever nutrients are needed from the extracellular fluid and bringing it into the cell. With phagocytosis, a lot of times we'll see this as in the immune system, which we'll go over later on in the class. But it's going to see like with your macrophages, there's going to be a bacteria out here, and this macrophage is like, hey, well, it's in the body, it shouldn't be, I'm getting rid of it. So it's kind of like Pac-Man. You're going to have this big macrophage, and it's going to extend until it wraps around this, and this will keep coming in, so that then digestive enzymes can be added to it. But phagocytosis is just eating. That's the best way, that's like the basic, simple, broken down version. Phagocytosis is eating something, whether it's cell debris or bacteria or honestly a lot of stuff. And then we've got receptor-mediated endocytosis, which is honestly probably one of the cooler ones. So, you're going to have your plasma membrane right here, and there's going to be all these little receptors on the outside of it. And when it needs something or needs to bring something in, whatever that thing is, will end up binding to each of these active sites. It'll bind to all those active sites, which will then end up like pushing in. So this whole thing, right? They'll bind to all those active sites. And then it's going to end up forming that vesicle like I talked about before. And it's got like whatever that nutrient or whatever is needed. And at some point, this is going to pinch off, and this is going to go into the cell. But receptor-mediated just means that whatever is needing to come into the cell, it's going to bind to a receptor before it can be brought into the cell and then used by the cell. Used by the cell. Okay, so that's your endo and exocytosis. Just remember that endo and exocytosis both start with an E, as does eukaryotic, so it's a strictly eukaryotic process. From there, let's talk a little bit about active versus passive transport. It's just like it sounds, active versus passive. Either passive is just going to kind of freely do what it wants. Active requires a little bit of energy. So with your passive transport, that's going to be things like diffusion and osmosis. It requires a small non-polar molecule. Because as we talked about with your plasma membrane, you're going to have a hydrophilic and a hydrophobic region, right? So you have your hydrophilic or water-loving head, and then you'll have a hydrophobic fatty acid tail. Now, with that being said, this has to do with like specific partial charges that we talked about earlier in the chemistry lectures that we had during the first test. But if you're having a molecule, like let's say it's small, but it's polar. These parts are polar too. So it's either going to get stuck at the head of the phospholipid or stuck within the middle of the phospholipid if it manages to get inside. So you're going to want to have small, non-polar polar molecules. And when you have that, they kind of just move as they see fit based on concentration gradient. So in high school, I know that a lot of students have done this where you take a semi-permeable membrane, you stick molasses in it, and then you stick it in water. And then you come back like 3 or 4 days later, and what used to have a lot of molasses in it is now extremely swollen. And we do have terms for that, but it'll be extremely swollen because the water rushes into it. Because water likes to follow a concentration gradient, where it goes from high concentration to low concentration to kind of even things out. So if you've got over here, we'll draw 2 little, like, let's say these are cells, right? Or just, okay, we got 2 things. And this one's gonna have like a lot of like salt in it. So let's say this one's salt water. Okay, that one's salt, you can't even see those. Okay. So let's say this one's salt water, right? And you've got a tube connecting these. And this one's fresh water. Obviously, fresh water isn't going to be completely free of salt, so there's going to be like a couple little salt things over here, salt molecules. But it's obviously got a lot more water molecules than this one does, because there's not as much space for all the water in this side, given all the salt molecules. So with diffusion, what's going to happen is the water from here is going to move through the tube to here to try to even out that gradient and make it so that this has a little more water and not as much salt. So you might see the water like the water level in this one rise and the water level in this one lower. That's the entire like gist of passive transport. However, with passive transport, when we're talking about like specific proteins, when we're talking about specific proteins within the plasma membrane, what those are going to be called is their channels. You've got pumps versus channels, which are the 2 like categories of proteins that we talk about a lot in this class that are within the plasma membrane. With your channels, things can kind of move freely. If you've ever learned a little bit about like action potentials, you've got your sodium and your potassium channels that can kind of be open and shut, but 9 times out of 10 they're open, and the potassium or sodium just can rush in and out as they see fit to make that membrane potential what it needs to be. So this is your passive transport. Now let's talk a little bit about active transport. Active transport requires energy, meaning it needs ATP. So with active transport, that's going to be your larger molecules, and you're always going to end up using those, like the protein that it goes through is always going to be called a pump, because it's got to pump it through. So with this, I mean, now it's erased, but with that, where it was going from a concentration of high, like from a high concentration of those salt molecules to a low concentration, or at least the water was, it's going to be the opposite. So with active transport, usually it moves against the concentration gradient, going from a low concentration to a high concentration, just to reach whatever potential is needed or to match the needs of that specific cell. So there's active and passive transport. However, we do need to talk a little bit with the passive transport about tonicity. So we've got our hypotonic, our hypertonic, and our isotonic. So, okay. This is like your normal cell, right? So we see this a lot with red blood cells and like why you have to put in a specific amount, like a specific concentration when you give someone an IV. So if you have an isotonic solution, that means that the concentration gradient outside of the cell is the same as inside of the cell. So water will just move back and forth like at the same rate. But if you put your cell in a solution that is hypotonic, that means that there's more water on the outside than there is in the cell. So all this water is going to be rushing into the cell, which will cause it to swell. And that can cause the cell to lyse. If you have a cell wall on it, it will just cause the cell to swell really bad, but it's really bad for the cells and can destroy them, as we saw, if you just have one with a plasma membrane, because it'll lyse the cell. But this is with your hypotonic solution, where there's a lot more stuff, right, on the inside of the cell than there is on the outside of the cell. So all of the water from the outside of the cell is going to rush into the cell, trying to make that concentration gradient like a lot more equal. So then we have hypertonic. And what happens here is you're going to have your cell, but it's going to be in a solution that's got a lot more water in the cell than there is on the outside. So then all of the water is going to rush out of the cell, which will cause it to shrivel. And you have looking like this, right? And we call that crenation, at least with red blood cells particularly. But all of the water is going to rush out of the cell, essentially like dehydrating the cell. So if you've ever like you see a grape, you see a raisin, they're made out of the same things. You just pull all of the water out of the grape to make that raisin. That's the same concept here, is you're taking the water out of the cell because the solution is hypertonic. Okay. Now, respiration. This personally is a very hard topic for me to understand, just because there's a lot of stuff going on throughout it. So I'm going to give kind of like a brief overview and like important parts of it. As for the specific process, like I said, there is a video linked below. Sorry, there's a video linked below. It's an amazing crash course by, I think, Kate Green. That's who doesn't. I personally love his stuff because he doesn't like dumb it down, but he puts it in a language that I'm able to understand, so that when I go look at the normal vocabulary, I understand it a lot better. So, with that being said, click the link to the video below, because what I'm going over right now is a very, very, very short, minuscule amount. But, okay, so we talked about oxidation and reduction. When it comes to like specific processes, this is kind of like another rule of thumb other than gaining and losing an electron, is that it's going to gain a hydrogen. So if you have NAD+, and you add a hydrogen to it to give you NADH, that's a reduction, because you're adding that hydrogen to it. Whereas if you were to go back this way, it would be an oxidation reaction. So, oxidation, you can do oil rig or Leo the Lion-Sesgur. I like Leo the Lion-Sesgur a lot more, because loss of electrons is oxidation and gaining electrons is reduction. Or oil rig, oxidation is losing, and then rig, reduction is gaining. So, either way you want to remember that. But if you're adding a hydrogen to it, that's also going to be a reduction reaction. And if you're losing that hydrogen, it's going to be an oxidation reaction. So, for carbohydrate metabolism, regardless of which way you're going, whether it's aerobic or anaerobic, you're always going to start with glycolysis, which is super important to understand. And that's going to produce very few ATP in the grand scheme of things. So let's get a little bit into anaerobic respiration, just because I've got a few little key points to add in there. So with anaerobic respiration, you don't have oxygen, right? Hence anaerobic. So that means you have to find a different way to help break things down. So you can go one of two ways. You can do lactic acid fermentation, or you can do alcohol fermentation. But that's entirely dependent on what you are.